So yeah, welcome everyone, and good afternoon. So this is the first of the ID-EPI seminar series. It's hosted by the Centre for Communicable Disease Dynamics and the Harvard C.H. Chan School of Public Health. Yeah, this is the first seminar. I hope to see a lot of you for the next few months as we have a great lineup of speakers. So I'm the moderator for today. My name is James Hay. And we're really excited to welcome Dr. Leah Katzelnick, who is a Stadman tenure track investigator and the chief of the viral epidemiology unit and immunity unit at NIH. So I'm just going to quickly run through some logistics. So the seminar is being recorded. So if you don't want to be in the recording, please turn off your camera. And the recording will be available on the website at a later time. We've got approximately 45 minutes or 40 minutes for the talk, and then 15 minutes for questions. So the way we're going to do questions, we're going to have everything at the end. And if you just send a direct message in the chat to Flavia Kompanova, she will be compiling the questions and we will go through them at the end. So Flavia, if you can introduce yourself, just say hi so everyone knows who to message.
Hi, everyone. Happy to take in questions.
Thank you. So we have Dr. Katzelnick today. So Dr. Katzelnick's research interests, they do cover a lot of ground. They range from antigenic and genetic viral evolution through to epidemiology and immunology and things like transmission dynamics. So I think it's going to be a really great talk. So we're thrilled to have her with us today, and she's going to be talking today about the immune interactions of Zika and dengue using data from a really fantastic pediatric cohort that's ongoing in Nicaragua. I think probably many of us on this in this talk will have seen data from that cohort over the years. So without further ado, let's get started. And thank you for joining us, Dr. Katzelnick.
Well, thank you very much for the introduction and for the opportunity to be here today speaking to you. So I am going to focus my talk today on some of the work we've done thinking about immune interactions between dengue and Zika viruses. One study that was published last summer, and then another study that is very complementary to that, which is following up thinking more about antibody dynamics after dengue and Zika virus infection, which we just submitted. So I wanted to start by just letting you know a little bit about my interests previously, as well as some of the work that we're going to be doing in the new unit that I'm leading at NIH. So today I'm mostly going to be talking about our immune correlates work. Just to confirm, you can see my cursor?
We can, yeah.
Yes, okay, good. So focusing on immune correlates, but I also have spent a lot of time thinking about viral antigenic evolution, as well as trying to translate immunity into population transmission dynamics. And some of the talk today will touch on those other themes. So the main approach that we will be using is to continue working with large longitudinal cohort studies and population-based studies, including the study in Nicaragua, which is led by my postdoctoral mentor, Eva Harris, as well as my other mentor, Dr. Angel Balmaceda, who supervised me when I was in Nicaragua working with the team. But we also work with other cohorts and we'll continue expanding those collaborations as part of their work. And the goal is to look at these large epidemiological studies using mostly assays for measuring B-cell immunity, using different serological assays, some standard, some new ones that we're developing in the lab to really start thinking about the determinants of protective immunity. So we'll start by talking about my favorite set of viruses, the 4 dengue viruses. Each of the 4 dengue viruses is capable of causing dengue, hemorrhagic fever, and dengue shock syndrome. You can see sort of the pyramid of the burden of disease, where somewhere between 2.5 and 4 billion people live in areas at potential risk of dengue. Only a small fraction of those are actually fatal, but dengue does cause a huge number of hospitalizations every year, and somewhere between 50 and 100 million dengue cases. This figure is also over here showing the distribution of the Aedes aegypti mosquito. Some areas shown here have active dengue transmission, others, like in the US, it's possible to have transmission. And dengue is one of the few infectious diseases that's actually become significantly worse over the course of the 21st century. And so it continues to be a problem. So one of the most interesting things about dengue is this mechanism called antibody-dependent enhancement. So this is where if you've had a first infection with one dengue virus, in this case shown here is dengue 1, you actually are at potentially increased risk of severe disease during your second infection. The first infection induces antibodies that don't just not protect you, but can actually increase disease severity. And the mechanism for this, antibody-dependent enhancement, this is a simple diagram of it, but the idea is that these heterotypic antibodies bind to the new, so heterotypic antibodies induced by the previous infection bind to the newly infecting serotype, aren't able to neutralize the virus and facilitate entry into myeloid cells via the Fc gamma receptor. And this can lead to both an increased number of infected cells and actually increased viral replication in those cells. And that this mechanism leads to the severe immunopathological events that are seen for dengue hemorrhagic fever, dengue shock syndrome. The figure here is just a kind of classic diagram of antibody-dependent enhancement, where you see a peak in enhancement at intermediate levels of antibodies, whereas you see lack of enhancement at lower and higher levels of antibodies. So there's sort of a sweet spot or range that is associated with the highest number of infected cells via this mechanism. So Zika virus was a little known virus up until quite recently. When the main virus taxonomy was published in 2012, it was barely even mentioned. It's shown here, close with the 4 dengue viruses. But as Zika emerged across the Americas, starting around 2014, 2015, it obviously became a disease of major importance, especially when it was shown that it wasn't just a minor dengue or a mild dengue disease, excuse me, but that in fact it could cause really severe outcomes, including congenital abnormalities and microcephaly in infants born to Zika infected pregnant women, as well as Guillain-Barré syndrome. So the 2 questions I want to focus on today are first thinking about immune interactions among the dengue viruses and whether or not these extend to Zika, right? So we know that there's this enhancing possibility for primary versus secondary dengue infection. Does this happen when one of those infections is actually Zika? And so in the first case, as Zika was merging, everyone was very worried that a prior Zika or prior dengue immunity might enhance secondary Zika disease. This has now been studied quite extensively, and I'll touch on that a little bit. But what I'm going to focus a lot of our talk on today is whether Zika infection can increase your risk of secondary dengue disease. The second question is very complementary to this, is saying, okay, well, let's say now we know that there is this relationship between prior pre-existing antibodies and risk of secondary disease, but are those antibody levels changing over time? And one of the main hypotheses for how immunity does change after primary dengue infection is that there is actually an initial period of cross-protection right after primary dengue infection, which is thought to last about 2 years. But these antibodies wane over time to not just not protective levels, but these dangerous enhancing levels, and are associated with your secondary risk of disease. Whereas after your secondary infection, you're thought to induce broadly protective antibodies, not just against the 2 viruses you were infected with, but all 4 dengue viruses. And so we're going to be revisiting that. And we measured the kinetics of antibodies long-term after different sequences of dengue and Zika infections, trying to test whether or not this is really the case. Okay, so just to return to thinking a little bit about immune interactions between dengue viruses, even though a lot of the papers on antibody-dependent enhancement were published in the 1970s and 1980s, and we thought this was an established done deal, the conversation was really reignited following the publication of the long-term follow-up of one of the phase 3 dengue vaccine trials. So shown here on the left is a scheme of the Dengvaxia dengue vaccine, which is just one of 3 main leading dengue vaccine candidates. This is the only one that's been licensed. And it's based on the yellow fever 17D vaccine, but the structural proteins, the pre-membrane protein and the envelope protein from dengue 1 through 4 have been substituted in. So this vaccine performed well in the first 2 years of the trial, but in the 3rd year, this really disturbing result came out that children ages 2 to 5 in the trial were actually at significantly increased risk of experiencing hospitalized disease. And in this trial, immunity hadn't been measured before for all participants before they received the vaccine. And so a major question was, is this an age effect, or is age here really just a proxy for immunity, right? Younger children are more likely to have not had dengue before. Was it that the vaccine was causing enhancement? At this time, the vaccine ended up getting licensed for older age groups in areas with higher seroprevalence, but whether it actually was causing enhancement had not been established. So we were interested in trying to look at enhancement in a human population at the same time, not in a vaccine study, but at least in natural infections, to try to revisit this question to say, okay, you know, is there really enhancement in human populations? So this is just sort of a scheme for the Nicaraguan pediatric dengue cohort study. Overall, it's followed over 9,000 children ages 2 to 17. The top is showing you the distribution of cases in the cohort for the first from 2004 to 2015, and the distribution of dengue ELISA titers. So naive means they haven't had a prior dengue infection. Less than 21 are low titers, and the darkest purple are the high titers. So we looked at how these pre-existing antibody levels were associated with the probability of experiencing the most severe form of dengue, dengue hemorrhagic fever, dengue shock syndrome, compared to being naive, and found that intermediate levels were associated with increased probability of disease compared with higher or lower levels of antibodies, consistent with the sort of trend of how antibody-dependent enhancement is supposed to work based on in vitro assays. This was measured with binding antibodies. A similar effect was seen when we looked at dengue with warning signs and severe dengue, although more moderated. And this is an expanded definition of severe disease that is designed to capture other forms of disease that aren't as specifically linked with classical, with the classical form of severe dengue, which is DHF-DSS. And you only really see protection when you look at any dengue disease. So we collaborated with Henrik Salje and a team who was studying a cohort in Thailand and observed similar effects as this in a completely independent cohort, and have followed up on this observation last year with Jesse Wagner, looking at viral load, showing that viral load is a statistical mediator of antibody-dependent enhancement of severe disease. So soon after these results came out, the Dengvaxia vaccine was shown to have this differential effect for seropositive and seronegative individuals. So individuals who are seronegative, so the children who entered the study who hadn't had dengue before, the vaccine significantly increased their risk of severe disease, and the effect was most pronounced when it was most similar to vascular leak syndrome and classic DHF-DSS. Whereas seropositive individuals, people who entered with at least one dengue infection before they received the vaccine, had protection. These results came out after they'd already started mass vaccination campaigns, including in the Philippines, and caused a huge controversy and backlash against vaccines, against the dengue vaccine, but also vaccines in general. So this is something to have in mind as we're thinking about other new vaccines coming up. So this is a problem for dengue, but a big question was, should we also be thinking about these possibilities for Zika vaccines? And so our work and work by many others, right, has shown that low levels of antibodies can enhance subsequent dengue disease, whereas high levels are associated with protection. As Zika was emerging, many researchers showed that prior dengue immunity wasn't necessarily associated with enhancement of Zika, although it really depends on your outcome. When you're looking at a population-based study of, a non-pregnant population, prior dengue infection seems to be, if anything, protective, but there's been some evidence showing that there might be an enhancement effect in maternal studies, but the sort of, this is still something people are continuing to follow up on. And we were really focused on Zika followed by dengue, this later piece, right? After this big Zika epidemic, what's going to happen when dengue returns? And there had been some studies in non-human primates showing that prior Zika infection could enhance viremia. when non-human primates were challenged with secondary dengue disease, but it hadn't been shown for humans. So we again returned to the cohort to test whether prior Zika infection histories could affect your risk of dengue disease. So it's the same cohort followed up, into this later period, right? So Zika came into the cohort in 2016 and caused an absolutely enormous epidemic, much bigger than had ever been seen before for any of the previous dengue epidemics. There were 2 years of very low transmission of dengue and Zika. And then in 2019, when I was working with the team in Nicaragua, actually in 2019, there was the biggest dengue epidemic that had been recorded over the entire period of the cohort. So we went back and tried to identify different dengue and Zika infection histories in the cohort during this period to look at how those were associated with the probability of experiencing dengue disease during this epidemic. We again used the dengue inhibition ELISA, which we had also used in our previous study of enhancement. This is just a scheme showing that you have your dengue 1 through 4 virions, you add your antibodies, your sample of interest, and you compete those sera with highly dengue specific antibodies that have been conjugated with HRP, and you detect how well the sample antibodies prevent the competition antibody from binding. This was an assay designed by Dr. Angel Balmaceda, and he also developed new assays as Zika was coming in, one using also a competition assay where antibodies compete for binding to the Zika virion with a Zika-specific E-domain 3 antibody, and a similar concept, but based on the Zika NS1 protein. And so using documented dengue and Zika cases and identification of inapparent infections based on these 3 assays, we reconstructed the infection histories throughout the cohort, where here you can see in gray naive. In 2016, when Zika came in, actually a large fraction of the cohort was naive. And so by 2019, a huge portion of the cohort had experienced a primary Zika infection only. Some had dengue and then Zika, others had multiple dengue infections before Zika, and a few had not been infected with Zika during the pandemic and retained just dengue infection histories. So we looked at how these different infection histories shown here on the x-axis were associated with the probability of experiencing first symptomatic dengue in the cohort, and found, as expected, that primary dengue infection was associated with increased probability of secondary symptomatic disease, but primary Zika infection was also associated with increased risk. And somewhat surprisingly, while sequential dengue infections are associated not with elevated risk of disease, it brings your risk back down to not being different from naive individuals. Individuals with a dengue infection followed by Zika infection were still at increased risk of symptomatic dengue. That second Zika infection didn't seem to provide the same protection as a second dengue infection. We were interested in also whether this was associated with more severe forms of disease, and again found that primary Zika infection as well as dengue followed by Zika infection were associated with increased probability of both dengue with warning signs and severe dengue, as well as dengue hemorrhagic fever, dengue shock syndrome. Overall, there weren't that many cases of dengue hemorrhagic fever, dengue shock syndrome in the cohort. And so to further follow up on this observation, we tried to then ask a similar question, but using a hospital-based study. So the Nicaragua hospital study has also been ongoing, in this case, since 2005. And in this study, children enroll at the point when they're presenting to the hospital. So usually days 1 to 7 post-symptom onset, and those that meet the criteria for dengue are invited to participate. This was expanded to include Zika and also chikungunya as they emerged in Nicaragua. So because we're looking at only their acute and convalescent phase sera, we needed to use a different assay for identifying their prior Zika infection histories. And so here we worked with Dr. Prem Kumar and Dr. De Silva at University of North Carolina, Chapel Hill, who developed an assay that they had found worked very well at looking at acute and convalescent sera. So this is based on the Zika E-domain 3 protein, and you test how well your sample antibodies bind to Zika E-domain 3 of Zika compared with dengue. And we confirmed that this performed well on samples from the cohort where we knew their infection histories, and then we applied these samples to the hospital study. So this is just showing the distribution of infection histories in the whole cohort study, which we've been talking about before, just among cases in the cohort, and then just among the hospitalized cases in this expanded subset of the hospital study we were looking at, where you can see that it was a larger fraction in the hospital study with the Zika histories. And we combined all of the cases in both the cohort and hospital study to look at the probability of experiencing a severe dengue case among all dengue cases given your different histories, comparing naive, those who only had evidence for a history of prior dengue infection, and those that had evidence of a Zika infection, some prior Zika infection with or without a prior dengue infection. And again, found that both those with prior dengue or Zika and dengue were at increased risk compared to naive individuals alone for both of the definitions of severe disease that we had used. So in addition to thinking about infection histories, we wanted to return to our studies thinking about pre-existing antibody levels and disease outcome. So this is just re-showing what we talked about earlier, where we showed that there was this specific range of antibodies associated with the highest probability of severe dengue. We reanalyzed the data using the same methods that I'm going to show you that we use for this study to account for repeat measures and to allow us to better compare between the our analyses from the single year in 2019 to all of the historical epidemics, and showing that you see mostly a protective effect against symptomatic dengue, where you really see this enhancing effect only for the more severe forms of dengue disease. So we focused on IELISA titers measured in 2019 against experiencing symptomatic and severe dengue 2 disease, and actually observed something slightly different from what we'd seen when we looked at our all serotypes combined in the previous studies. We found broad enhancement of dengue 2 disease, including symptomatic dengue, with this kind of enhancing effect narrowing as you go to the more severe forms of disease, right? It really narrows to this range of low to intermediate titers 21 to 80. We also looked at Zika titers measured at the same time point using the Zika inhibition ELISA, and also observed these similar patterns of, broad enhancing effects across antibody titers for symptomatic dengue, and it becoming narrower as you look at more severe forms of dengue disease. So we had found some evidence that a prior Zika infection can enhance secondary dengue 2 disease, but we were interested in seeing whether this different pattern of broad enhancement that we saw against dengue 2 was something about prior Zika infection, or is this something about the secondary infection being caused by dengue 2? So we went back and looked at, in the previous years of the cohort, how prior dengue infection affects your risk of dengue 2 disease. So here again is showing what I just showed you, antibodies in 2019 against dengue 2 disease. Going back, we also found that there is some broader enhancement of symptomatic dengue disease when the secondary infection is dengue 2. And again, this narrows to severe disease, although it's not significant. When you're just looking at dengue 2, there just weren't enough cases then. So this was interesting. Okay, so why do we see something different from dengue 2 than we did for, enhancement overall? So we looked individually at how prior dengue infection is associated with risk of dengue 3 disease, separately for dengue 1, and then also thinking about dengue in relation to Zika secondary infection. So looking back, we see that prior dengue pre-existing antibodies against dengue 1 are associated with mostly protection against symptomatic dengue 1. And there really isn't much evidence for enhancement of dengue 1, even when you're looking at the most severe form of disease. Dengue 3 is somewhere in between, where you see both an enhancing and a protective effect for symptomatic dengue that really narrows again to the enhancing effect when you're looking at severe disease. Whereas when you're looking at antibodies in relation to secondary Zika infection, we mostly see a protective effect. So pulling all of this together, we see different patterns of the relationship between prior immunity and your secondary infection outcome, depending on what the secondary infection is. But we see some similarities depending on, kind of for both prior dengue immunity and prior Zika immunity. So we saw enhancement of dengue 2, protection and enhancement of dengue 3, protection against dengue 1, and protection by prior dengue and Zika immunity against secondary Zika infection. So we might hypothesize that these interactions would be symmetrical in some way, right? That prior Zika infection might also be more protective against dengue 1, protective and enhancing against dengue 3. And dengue 4, we don't know, we didn't observe that at all in Nicaragua, but maybe based on what's been published previously about interactions between the dengue viruses, we might expect that dengue 4 might also be enhanced. So this is, again, a hypothesis. We don't have any idea how this will hold up, and it really needs to be studied in other populations where they've observed these sequences of infection. So just a few comments on Zika vaccines. So one concern is that monovalent Zika vaccines that induce cross-reactive antibodies like those that we're seeing after natural Zika infection may be able to increase risk of subsequent symptomatic and severe dengue disease. However, the good news is that many Zika vaccine developers have been thinking about this from the beginning. This was always a concern, and have been trying to design Zika vaccines or Zika vaccination strategies that are less likely to be able to enhance dengue. And that this isn't even just different depending on the overall approach that you're using for vaccine, but even within different vaccine constructs within the same platform, there can be variation in terms of the antibodies that are being induced. So hopefully this won't be a problem for Zika vaccines, but it's something to keep on the radar. So thinking about our second question that I posed at the beginning, so how are antibody levels changing after infection, right? So we showed that pre-existing antibody levels are associated with your risk of secondary disease, but how do you actually get to that level of antibodies, right? Are they changing over time? So we have returned to this hypothesis that antibodies wane in the period after primary infection, and that this waning over about a 2-year period is what explains the epidemiological observation that you usually see about 2 years between a first infection and a secondary symptomatic or severe infection, as well as many different modeling studies that have shown that you kind of need some amount of waning of immunity to explain the epidemic dynamics of the 4 dengue serotypes. So our hypothesis was that cross-reactive antibodies would wane for about 2 years after primary infection, but would be stable after secondary infection and explain the long-lived protective immunity against secondary dengue disease. So here we looked at antibodies measured longitudinally in both the hospital cohort as well as the pediatric community-based cohort. We used both generalized additive mixed models and linear models for looking at antibody titers. And we're focused on measuring both group and individual level antibody kinetics, so half-life, as well as the magnitude of antibodies, generally written about as either magnitude or set point. This is just a nice conceptual plot from Rustin Antia's analyses of longitudinal responses after vaccination. And again, we're looking at the same infection histories that we were looking at in the previous study and using the same 3 assays, the dengue inhibition ELISA, Zika inhibition ELISA, and Zika NS1-BOB assay. So we first looked at immunity after dengue infection and focused on anti-dengue antibodies, so measured with the dengue ELISA. And quite surprisingly found that antibodies were very stable. Initially in the hospital-based study, there was a period of antibody growth, likely associated with affinity maturation of B cells after a first infection, which we observed peaked around 3 months. There wasn't a 1-point time point, so it's possible that the peak was actually earlier. That waned again by about 8 months, and then we're very stable thereafter. And in the community cohort, we looked at it out to up to 11 years and found sort of an astonishing amount of stability. So this is just showing the distribution of half-lives where you see this period of growth in the hospital, but then long-term stabilization between 8 and 18 months in the hospital study, which is how many is the longest we have from the hospital study, but out to many years in the cohort. And about 50% have titers above 80, right, which we've shown previously are associated with lack of enhancement, and in some cases, if it's high enough protection, and about half are within a range that's associated with potential enhancement of future disease. After secondary infection, we observed rapid antibody decay initially, which isn't very surprising. You experience a huge restimulation of prior immunity during your second infection, plasmablast production that induces a huge number of antibodies that wane quite rapidly. And we found here that they wane rapidly initially out to about 8 months and then begin to stabilize and reach their long-term kinetics. But somewhat surprisingly, in the cohort, we found that there was some evidence for longer term but very gradual decay. This was subtle, and some individuals did have stable titers, but a fraction continued to decay at a slow rate over many years after secondary infection, which was surprising. So this is a contour plot just visualizing the half-life and titer magnitude for children in the cohort, shown for primary dengue, those with exactly 2 dengue infections, and those with multiple prior dengue infections, where of course those with multiple prior infections have higher titers, but they also have more rapid decay. And even though the majority were expected to maintain protective levels for at least 10 years, a sizable fraction were predicted to have titers drop into this enhancing range, so below 80 within about 5 years. It's hard to look at whether they stabilize longer term. We have some evidence that the titers don't really drop below the levels they were at after a primary infection. They generally seem to drop to the level observed after primary infection, but we don't really have enough data to prove that definitively. So this just raises the question of whether there's an importance of re-exposure in endemic areas for maintaining high levels that are seen after secondary infection for some individuals. So we were also interested in understanding if there was also maybe decay after a Zika infection. So dengue transmission was surprisingly low across the Americas after the Zika pandemic. This is just showing you the figure I've shown you multiple times before with the big Zika epidemic in 2016 and this incredibly low transmission in 2017 and 18, right before dengue emerged. Others really focused on this observation. This is figures from Becky Borchering's paper from a few years ago showing where they followed dengue for many years in Brazil before the Zika pandemic and after, and showed that there was a decline in Zika as well as in Brazil and in Colombia. Others have been following up on this as well. And so the hypothesis was that Zika initially induced cross-protective antibodies against dengue that waned over time, and then individuals became at risk of severe dengue disease. So we hypothesized that we would see something similar to what we thought would happen after primary dengue. But instead, we observed that anti-dengue titers actually slightly increased over time after primary Zika infection. So here we're looking at children who experienced a primary Zika infection and their cross-reactive titers to dengue. And instead of waning, they actually increased. When we went back and looked at heterotypic neutralization titers in a small subset of children that we've looked at in the dengue cohort after dengue infection, we also have seen a similar pattern where titers can increase for a period of time after primary dengue. And there have been similar observations of dengue and Zika memory B cell responses, not over years, but over many months that have been published previously. And other people who have hypotheses for what could explain this, I would be happy to discuss that in the questions. Another is that there is some possibility that there's a bias because these were low levels that were more likely to detect increases than decay because of the limited detection of our assay. But certainly we're not seeing strong evidence for waning. If anything, titers are increasing. However, after secondary infection with Zika, so individuals who had dengue followed by Zika, we do see waning, and that it looks very similar to what was observed after secondary dengue infection, where you see an initial period of rapid decay followed by stabilization. Of course, we don't have as much data or as many years of follow-up for this group because the Zika pandemic was so recent, but the overall pattern is similar. And finally, we were interested in thinking about not just how anti-dengue antibodies after Zika, but anti-Zika antibodies after Zika and what their kinetics are. Again, comparing between primary Zika infection and those with secondary Zika. So one dengue infection followed by Zika or multiple prior dengue infections. And what was interesting is when we looked at the Zika ELISA, which are virion-specific antibodies, or NS1, we saw similar patterns after both primary and secondary Zika, but it differed by antigen, right? You see different kinetics against the virion compared to what we've seen for the NS1 protein. And this is somewhat consistent with studies showing that the Zika-specific response after Zika infection, regardless of your prior dengue response, is really largely induced by naive B cells, even when you've had prior dengue. There are cross-reactive antibodies that are induced, but those aren't what have been shown to be responsible for the potent anti-Zika response. And this is from not our study, but many studies in the literature, both looking at memory B cell responses as well as serum antibodies. So a few conclusions from this, we find that the antibody kinetics are really different depending on when you're looking at primary versus secondary infection, the specific virus, the antigen, and also there's huge heterogeneity across individuals with set points that we really see after primary infection, which seem to be related to your probability of secondary disease, not antibody waning across individuals that is associated with risk. We find that the antibody kinetics that we look at find in individuals don't directly map to what's been described for population level transmission dynamics. And this isn't so much of a problem because there's many models that have already shown that you don't need 2 years of waning to see a 2-year gap between epidemics, and also some really nice work looking at other factors that can lead to a 2-year gap between sequential infection with different serotypes, including looking at spatial heterogeneity. And finally, that there may be a value to looking at antibody responses measured about 8 months after infection, and that those are potentially really useful for the long-lived antibody response. Often in vaccine studies, titers are measured about 1 month after the final dose, and that might be too early to really represent the long-term response. So just briefly, I've really focused on the immune correlates portion, sort of the first theme, but the group is also going to be thinking about mechanisms of viral genetic and antigenic evolution, mostly for dengue and Zika viruses, but we're also discussing potential projects in this area for coronaviruses, as well as continuing to try to think about how we can use these deep measures of immunity from cohort studies to inform vaccine design and policy and thinking about transmission at the population level. So with that, I really want to highlight the work of the Nicaragua Pediatric Dengue Cohort and hospital studies. These are the co-authors on both of the studies. And again, thanking Eva Harris, my postdoc mentor, as well as the entire team in Nicaragua. And thank also our other collaborators. This is only listing some of the main collaborators for each of the cohorts that we work with, and the new viral epidemiology and immunity unit team, Ana and Fernando. We have 3 new members joining, and we're actively looking for postdocs working in epidemiology and infectious disease modeling to join us. So if you're interested, please reach out. And with that, I'll take any questions. Thank you.
Thank you so much, Dr. Katsanek. That was an absolutely beautiful talk. It's really, really fascinating to see the interaction of all those within host processes manifest themselves in the population. Like super, super cool. So we have almost 20 minutes actually for questions. So if you want to send those directly to Flavia in the chat box, that would be great. And we'll try and get through as many as possible in the time. So I'm just going to read from the questions now. So let's see. Okay, so the first question is kind of public health focused question. So how do you think these findings will impact future immunization strategies? Would people need to be tested for previous flavivirus exposure before receiving the vaccine? If so, are there rapid diagnostic tests that will make this logistically easier?
Yeah, sorry, I was going to talk about it and then I cut it because I was trying to keep to 40 minutes, but this is great. Now I can talk about it. So yes, in 2018, WHO changed the recommendation, as the person asking the question is probably aware, to say that Dengvaxia should only be given to dengue seropositive children. They made the recommendation to use rapid diagnostic tests, but at the time they made this recommendation, none of the rapid diagnostic tests were designed to detect historical infections. All of them were designed for acute or very recent infections within the last month. And so something that the vaccine developers, Sanofi Pasteur, has been following up on is trying to evaluate different existing rapid tests to be used for this purpose. We've also been studying this a little bit. So this shown on the right is an example of one of the rapid diagnostic tests. And most of the tests are read at 15 to 20 minutes. And this is shown here on the far left, where we looked at individuals who've had only dengue, or who are dengue naive, have had a primary dengue infection, or have had secondary dengue. And when you look at the first time point, it is a very specific assay. Almost nobody is called positive. So that's something the vaccine developer is hoping for, because that rules out vaccinating anyone who'd be put at increased risk of disease. But it also fails to identify almost all individuals with primary dengue immunity, who are the ones that are likely at highest risk of severe secondary disease, and only detects some fraction of those with secondary dengue immunity. So yes, currently we need to identify individuals who've had a prior dengue infection. I haven't even shown here the issues of prior Zika infection histories, where, you know, the current idea is that you need to be able to identify those only with prior dengue immunity and not prior Zika immunity. That's a whole separate, even further complication. But we need better tests or better ways to use existing tests to do this. Otherwise, we're only really going to be vaccinating individuals who already have very high levels of pre-existing dengue immunity. And there's still a value to that. The vaccine developer has shown that that still provides some protection, but it isn't protecting the vast majority of people who are at risk.
Great, thank you. I actually have a quick question, or hopefully quick. Most of the results you've shown have been in pediatric cohorts, and I don't really know about the epidemiology of dengue enough to know if this is a stupid question, but could you speculate on the role of these mechanisms in adults, for example, as individuals get older and maybe there's an immunosenescence of their memory B cell response that might put them back in the risk zone?
Yeah, that's a great question. Dengue has largely been studied as a pediatric disease because the most severe disease is usually seen in pediatric populations when transmission intensity is high. And so it's very hard to disentangle the relationship between transmission intensity in a population, right, which is associated with age, as well as overall age effects, right, which is given that you're experiencing a secondary dengue infection when you're 1 versus 10 versus 50, right? Do you have different disease? And those are things that people are actively studying right now to try to understand different age effects. Even within a pediatric cohort, it does look like there's differences in age. The kind of severity you experience when you're very young is different from when you're older. I'm sorry. But this is something that only a few places can really study. So Singapore has a larger fraction of adults who get severe dengue disease, and now there's a lot of work looking at comorbidities and severe dengue disease. So it's an active area of research. Some of the same pattern of immunity, where if you're experiencing your secondary infection, regardless of age, it's associated with increased risk, but it does depend to some extent on age. I do have one figure looking at, age and some of the enhancement effects that we had looked at. And you actually see that this is age 4, age 8, age 12, and age 16. You see stronger effects with older ages, whereas weaker effects with younger ages, at least for the Zika, how Zika is associated with future risk of dengue disease. But it's a really important area to continue studying in adults.
Thank you. So the next question, I think, is related to antibody kinetics, or kind of peripheral to antibody kinetics. So the question is, do you think the time between this primary and secondary infection affects the risk of severe dengue and the antibody level? Thank you.
I mean, that was really the hypothesis we were trying to test. we don't really find that antibodies wane very much after about 8 months. So our hypothesis based on this is that individuals reach different, let's see if I can just go back to our model. I often show an updated version of this model where levels are stable. But the idea, our new hypothesis is that individuals reach a stable level of antibodies by about 8 months after infection, which is associated with their future risk of dengue disease. But that period in between 8 months and 2 years, right? 2 years is when we think you start to see increased probability of severe secondary dengue, is really related to your probability of being exposed to a new serotype. We know that there's spatial clustering of dengue cases, so that if in your own community, you're less likely to have a new serotype come in within that time frame. So while it's possible that you can get disease, you're simply less likely to be exposed to a new serotype. This also, to some extent, matches some of the dengue vaccine trial results, where at about 8 months, the Kaplan-Meier curve slopes really start to change about 8 months after the last vaccine dose, where you start to see the enhancing effect, not really 2 years later. So yeah, our hypothesis is that there's actually stable set points rather than this longer term waning after a first infection, which had been described previously. However, it's still important to look long-term in vaccine studies out to 2, 3, 4, 5 years to see enhancement effects. This isn't an argument that you can stop the trials earlier. You still need to look at it, especially to be able to see high enough numbers and detect if there is a significant enhancement effect.
Thank you. So the next question, I think, is related to finding out a bit more granularity about the cases in the first data set, I'm assuming. Sure. So the question is, what's the proportion of cohort/hospital cases that were due to primary, secondary, and tertiary infection?
Proportions of cases.
So
yeah, go ahead.
No, I think I should just clarify. I think that's right. The slide you're on is the
yeah.
Just to clarify that that plot on the right.
Yeah. So it's a combination, right? This is the total study participants, so the entire cohort and their distributions of dengue and Zika infection histories. The group that we are most powered to be able to detect disease in is the primary Zika group, of course, just because there's so many more of them in the cohort, whereas some of the dengue histories, we're getting into pretty small numbers by 2019. There just aren't that many left that only have those histories. This is showing the probability within those groups of experiencing symptomatic dengue, right? So you can see that it's almost 12% of the primary Zika group, which already formed a large fraction of the cohort, that did experience symptomatic dengue. It's smaller numbers once you're looking at very severe disease, because there just weren't that many severe cases, but it was still a pretty sizable fraction of each of those populations, especially for the primary Zika group. And then the next highest power group is the dengue Zika group, and yeah, the dengue histories, there just aren't as many of them. So it's a smaller number.
Thank you. So the next question is prefaced by amazing talk. So congratulations. I may have missed it, but you mentioned about the 2-year cycles between the severe outbreaks mentioned here. What about the immune response drives this on top of susceptible demographics/geography? A bit of a broad question, but how broadly generalizable is this across human viral pathogens?
This idea of so I don't know if the person can clarify, but this idea that these individual immune dynamics might not directly map to the 2-year dynamics for transmission?
I think it's, delineating the role of the immune response and what's going on at the geographical and demographic level.
Yeah, this is something where I'm hoping outstanding infectious disease modelers who are better at playing with and developing these kinds of models can help think about this. We've thought about it a lot because the epidemiological observation is that there is this 2-year gap between, I'll go to a few of these slides. So there's a 2-year gap between a first infection and a symptomatic secondary infection. There's also data both from other cohorts and even from our own cohort that there's, this is from a Thai cohort, this is from Henrik Salji's, had published this back in 2012, looking at the distribution of homotypic cases. So virus, infect cases, this is DHF cases caused by viruses of the same serotype versus a different serotype. And found that there was about a 2-year gap to heterotypic cases in the same area. They are also looking at spatial clustering. And this sort of 2-year temporal dynamic was again related back to, there's individual level immune waning, antibody waning. We also saw this 2-year gap in the Nicaragua cohort, where you see many more homotypic cases in the first few years and heterotypic cases after 2 years. Our main hypothesis, as I've been saying, is that there are other spatial factors that can really drive the distribution of diseases that aren't always accounted for in some of the historical models of dengue transmission, especially those that assume homogeneous mixing. So this is a paper from Mario Recker from also many years ago, looking at how you could get realistic dengue dynamics just by modeling spatial heterogeneity, by incorporating spatial heterogeneity in your model. He showed that you can also have waning of immunity, and that an enhancement can be incorporated in, and the kinetics are still, consistent with realistic dynamics, but that you don't need waning to produce realistic dynamics. Other models have shown that you only really need about a 1-year period of waning at the individual level to get 2 years between epidemics. And this has been shown in a few different models of both dengue and Zika epidemics. So I would love for anyone who has feedback to reach out to me if they have other ideas of how this works, and other hypotheses about how to take this to the next level, and if this is seen in other diseases.
Thank you very much. So we have 5 minutes. I'll ask one more question unless we get another few come through. So this is a very similar question. So in the cohort study, it seemed that during the Zika outbreak, the incidence of dengue infection was very low, and you proposed some immunological mechanisms for why that might be. Could you speculate on, are there any ecological reasons why that might be?
This is sort of one of the golden questions, right? Because we don't really see that antibodies wane after primary Zika infection. We do see that they wane after secondary infection. So one hypothesis is that a huge number of individuals, adults and children who'd already experienced many dengue infections got Zika. Zika dramatically boosted responses to dengue. This is another immunological mechanism, but that this provided some period of cross protection because of boosting immunity in adults, not actually primary Zika in children. Ecological factors, there could be interactions in the mosquito, although other people are much better suited to talk about that. I know some people were thinking about competition in mosquitoes and their ability to transmit one arboviral disease versus another, non-virus specific or adaptive immunity specific responses, but other immunological mechanisms that are more broadly antiviral. I don't have any other great hypotheses at this time if there's other ecological factors that have been driving this. I mean, it was a dramatic effect that was seen across the Americas, not just in one country, but in many countries. And so again, please reach out if you have other hypotheses about other ecological drivers that could help to explain it.
Awesome, thank you so much. I think we can start closing off the meeting there. So let's do a virtual round of applause for our speaker yet again. So that, I guess, concludes our first seminar of the series. So just on behalf of the Center for Communicable Disease Dynamics and the Harvard T.H. Chan School of Public Health, just want to thank Dr. Katzelnick again for joining us today. And thank you all, to all of you for tuning in for the first seminar of the series. Hope hope to see you in the coming months, and to remember to please check the CCDD website for the upcoming seminars and other events. So yeah, thank you very much everyone for joining, and have a good afternoon.